Okay, welcome back, everyone. We are going to be covering tissues today, the 4 different types of tissues. All right, so let me do a screen share. Let's bring up the PowerPoint on the 4 different types of tissues. Okay, so the 4 types of tissues. Remember, we said that chemicals come together, chemicals make cells, cells make tissues, tissues make organs, organs make systems, systems make the organism. Cells come together by different types of junctions and make tissues. The 4 primary types of tissues are epithelial tissue, connective tissue, neural tissue, and muscle tissue. Muscle tissue and neural tissue is the easiest. We can knock that out pretty quick. Epithelium and connective take a little bit more time. So remember with muscle tissue, it's just 3 classifications, right? Cardiac, smooth, skeletal. Neural tissue, we'll primarily be talking about the neuron, and that's pretty straightforward, the different parts of a neuron. Epithelium and connective tissue, you heard me talk about that when I covered blood. Remember I said when you look at blood, blood is connective tissue. Connective tissue has cells, but lots of ECM, lots of extracellular matrix. And that's the characteristic to connective tissue. You have some cells scattered here and there, like a few red blood cells and a few white blood cells, but lots of plasma, lots of extracellular matrix. Epithelium, on the other hand, is different. This is when you have a bunch of cells closely packed together with very little extracellular matrix. Epithelium likes to be on the surface, like around the skin. Epithelial tissue likes to line tubular systems, like blood vessels is a tube, intestines is a tube, the respiratory tract is a tube. So we'll see it a lot in the tubular systems. Epithelium tissue has a very specific way of classification, and we'll go through how you can identify that. All right, so epithelium, connective, nervous tissue, and muscle. The tissues have distinct structure or patterns, and when you look at different structure, they're all going to have different functions. To perform specific bodily functions, tissues are organized into organs like the heart, the kidneys, the lungs, and the arrangement of tissues in an organ are going to determine the organ structure and function. Remember, the histology is the study of structure and function of tissues. The person who studies them are called histologists. If there's any pathology to the histology, we call that pathophysiology. Epithelium, it likes to line and cover tissues. It likes to cover the external surfaces of the body, like the epidermis, that superficial layer of the skin is epithelium. It happens to be stratified squamous epithelium. And as I said earlier, they like to line the tubules or body cavities. Glands are classified as epithelium, and they're classified as epithelium because they develop from epithelium membranes. They protect us. They're used for absorption, filtration, excretion, secretion. Protection, we have the skin, right? And the skin can prevent us from type of abrasions or excessive friction. Because of the skin, things may not invade us like bacterias, but if there's a 3rd-degree burn and that skin is not there, bacteria can certainly evade. The skin and epithelium prevent us against chemical damage. We have cilia, which are these little hair-like structures on epithelium. We find them in the respiratory tract. I showed you that earlier in the trachea, how smoking paralyzes and destroys those cilia. And whenever epithelium becomes damaged, that epithelium morphs into another type of epithelium. They call that metaplasia. When one tissue type dies and morphs and evolves into a different type of tissue, we call that metaplasia. Metaplasia is usually precancerous. So we used to, or should have, pseudostratified ciliated columnar epithelium in the trachea and in the respiratory tracts. But when those cilia start to become paralyzed and die, we don't have pseudostratified ciliated columnar epithelium anymore. It changes into a different type of epithelium that becomes precancerous. We need the cilia to help sweep the dust and foreign particles away from the lungs. Otherwise, you get that, right, that smoker's cough. Epithelium is needed in the stomach, in the small intestines, where we have a lot of absorption of nutrients. We have epithelium in the kidneys. The kidneys are lined with epithelium to absorb things, such as water. We can secrete things, we can get rid of things. And the kidneys can also filter. So it cleans and returns something rejuvenated back into the system. It filters out the blood and pulls out the impurities and returns clean blood back into the system, specifically in an area called the nephron. The nephron of the kidney has many tubules. They're called kidney tubules. And whatever we want to get rid of, we can excrete. Glands can secrete. They can secrete their oil secretion, or sweat is a secretion, or mammary glands can secrete milk. And there are sensory receptors. So the epithelium that covers the body's surface contain many sensory organs. Okay, we have little hairs that have these hair root plexus associated with them. We can pick up deep pressure and light touch and pain and temperature. Cilia are long, slender extensions of the plasma membrane, and they help to move secretions and fluids across the surface of the cell. And microvilli are very, very small projections. Microvilli are more for absorption of nutrients. There's lots of microvilli on the lumen of the intestines, especially the small intestine. All right, so how do we classify and identify epithelium? So the first thing we have to do is we have to ask ourselves, when we're looking at it under a microscope or on a slide from the side view, do we see something that is 1 cell layer thick, or do we see something that is 2 cell layers thick? If we look at something and it's 1 cell layer thick, we say it's simple epithelium. So if we have this, if my hand is flat and it's very irregular shape, I'm going to call this a squamous cell, and it's 1 cell layer thick, I'm going to say it's simple epithelium. Once I add another layer to it, now it's stratified epithelium, stratified squamous epithelium. If I make it I'm trying to see if I have something here that I could use. If, let's say, this is a cube, this is a cube, this is a cuboidal cell, and if it's only 1 cube, 1 cell layer, let's say like this, 1 cube, it's going to be simple cuboidal. Once I add another cube-shaped cell on top of it, it is stratified cuboidal. If I have a cell that looks like this, like a column, and it's only 1 cell, it's simple columnar epithelium. If I put another column on top of it, now it's stratified columnar epithelium. Okay, so we have to first ask ourselves, is it 1 layer, then it's simple. If it's more than 1 layer, it's stratified. And you always have to look at the apical layer. The apical means the apex. What's at the very top? Not at the bottom, not at the basement membrane, but at the surface, at the top. Then again, you look at the shape of the cells. If it looks very flat and irregular, it's squamous. If it looks like a cube or a square, cuboidal. And if it looks like a tall building, then it's columnar. All right, so squamous, again, is a regular shape. They're flat. Their function is for protection, secretion, and diffusion. Diffusion means gases can diffuse, like oxygen or CO2 can diffuse from one side of the membrane to the other, diffusion. Cuboidal, like cube shaped, these are found in the kidneys, in those tubules, the nephron of the kidneys, and they're involved with absorption and secretion. So these cuboidal cells are found at the proximal convoluted tubule. So if you Google or look in your book and you look up nephron, you will see some structures like a glomerulus, a Bowman's capsule, a proximal convoluted tubule, and a distal convoluted tubule. The proximal, there's that word proximal at the beginning, the proximal convoluted tubule is loaded with cuboidal cells. And then you have columnar, which is, we said, is column shaped. Okay, pseudostratified means that it looks, pseudo means false. So it's not really stratified, but it looks like it's stratified because there are many columns, but they're really simple. It's one cell layer, but because the nuclei, the nucleus of each columnar cell may be at different levels, it gives the appearance that it's stratified, right? If all the nuclei are at one level, it's easy to see that it's simple. But if one nuclei is here and another one is here and another one's here and another one's here and another one's here, it looks like it's stratified, all right? Pseudo-stratified. Transitional epithelium, this is found in parts of the body where the parts may get larger and smaller. So the uterus could be one size or it can increase in size. The urinary bladder can get larger or smaller. The uterus can get larger or smaller. So that's transitional. Transitional is typically only found in the uterus or the urinary bladder. So again, when it comes to epithelium, we have simple epithelium that can be classified as simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium. In the stratified, you got stratified squamous, stratified cuboidal, stratified columnar. Then you have the two funky ones, pseudo-stratified. It's not really stratified columnar. It's pseudo, it's false. It looks that way. You see them a lot in the respiratory tract. And you can see them in certain parts of the digestive tract as well. And then there's transitional that's found at the uterus that can get larger and smaller, or the urinary bladder. So this, because it gets larger, right, the cells can stretch and change shape. It's not just one particular size and shape all the time. So here we've got simple squamous. You have one cell layer thick. Here it's simple cuboidal, it's one cell layer of cuboid cells. Here it's simple columnar, it's one cell layer of the column shape. Notice that the nuclei are at one level. But if we go down below it, the pseudostratified columnar, you can see it's only one cell layer, but if you look at the nuclei, they are scattered at multiple dimensions at different levels, so it looks like it's stratified, but it's really not. The cuboidal shaped, here now you have two levels, 1, 2. These two levels, now we have to call it stratified compared to simple. And here's stratified squamous, right? You go to the apex, you go to the top, and you could see that there's 1, 2, 3, at least 4 different levels here, and it's stratified squamous, okay? So that is epithelium. When we look at connective tissue is now when you have some cells. Let me go back for a second. Here, notice the cells are very closely packed together with very little extracellular matrix between it. But when we look at connective tissue, it's the exact opposite of that. We have some cells, but lots of extracellular matrix, okay? The cells are widely spaced with lots of matrix. It's the most abundant and variable type of tissue in the body. It's found all over, but it connects organs to each other. It even connects to the epithelium. It gives support and protection to us because blood is connective tissue. It's called liquid connective tissue, so it's part of our immune system. Fat or adipose is connective tissue. Fat is storage of energy. All fat is you can have a storage facility for sugar. That's a fat cell. The more sugar you have without energy expenditure, that fat cell increases. That's called a triglyceride. Fat or fat cells can help insulate. They're good for heat production. And connective tissue can move or transport materials through the body, such as blood. Okay, in terms of loose connective tissue, areolar connective tissue is a type of loose connective tissue that separates. Like if you pinch your skin, right? If you pinch your skin like this, you're only pinching skin. The areolar is what separates the skin from the muscle, okay? Adipose is loose connective tissue. It's the fat, okay? I wouldn't worry about reticular. I'm not going to test you on that. Dense connective tissue, there's dense regular and dense irregular. Dense regular connective tissue is when you have connective tissue that you have tons of collagen fibers that are closely packed together in alignment, okay? When the tissue is scattered in different directions, like in the skin, your skin, you can manipulate your skin in any direction. You can massage it in so many different directions, it's not going to tear apart because the fibers are in multiple dimensions. Elastic connective tissue, certain ligaments, especially of the vertebral column, are elastic. We have some ligaments that have more collagen, more protein for strength, and another type of protein called elastin for its elasticity. So the vertebral column needs much more elasticity. There's fluid connective tissue like the blood. Red blood cells, white blood cells, and platelets are all part of blood. There's lymph, which is the interstitial fluid found between the cells of the human body, and it enters lymphatic vessels. Okay, that's still liquid connective tissue. Cartilage is supporting connective tissue. We find cartilage between bones. The hyaline cartilage is the most common type of cartilage. We find it between diarthrodial joints. When we talk about bones, joints can be either a synarthrosis, amphiarthrosis, or diarthrosis. Synarthrosis is not a movable joint, and amphiarthrosis is slightly movable, but diarthrosis are freely movable joints. Your fingers are freely movable, the wrist, elbows, shoulders, hips, knees, ankles, feet, all freely movable. So between this bone and this bone right here where it bends is going to be hyaline cartilage. Between my wrist is hyaline cartilage. At my hip joint, there's hyaline cartilage. It helps to reduce the friction between two adjacent surfaces. Elastic cartilage allows for more flexibility, and fibrocartilage is the strongest, toughest cartilage in the body. The intervertebral discs, or the IVDs, intervertebral discs, the cartilage between your vertebrae, is fibrocartilage. Where the pubic bones come together, there's a vertical disc called the pubic symphysis. That too is fibrocartilage. Bone is connective tissue. And we spoke about bone before. We showed you the osteon, all of those concentric lamellae. Between osteons is the interstitial lamellae, which we said are remnants of other osteons. Osteoblasts are the immature cells that produce matrix. They're the bone-forming cells. Osteoblasts, when they harden in their own matrix that they produce, become osteocytes. The space that they're contained in is called a lacunae. Osteocytes are mature cells. They help maintain bone. An osteoclast is a macrophage. It dissolves bone. It breaks down bone. So the osteoblast with the B builds bone. The osteoclast breaks it away. Concentric lamellae and circumferential lamellae we've already covered. The canaliculi are those finger-like extensions, those channels that you can see in the concentric lamellae. They provide passageways through the solid matrix for the diffusion of nutrients and wastes. Volkmann's canal, this canal runs at a right angle to the long axis of the bone. It's kind of how the blood vessel, right? That blood vessel perforates, that nutrient artery breaks into the bone, but nerves can do it too. The periosteum is the outside of the bone, and the endosteum is the innermost part of the bone. The lacunae we covered, the periosteum we covered, the endosteum we covered. The endosteum is going to cover the spongy bone. It's close to the trabecula, trabecula is when bone lays down metrics, but it looks like a sponge. Compact is very, very different. The Haversian canal or central canal is where the blood vessels, the nerve, the lymph travels through the center of the osteon. So here is an osteon. Each of these is an osteon. Right there in the center is the Haversian canal. And we have these concentric rings going around it. And this is the spongy bone right center here. So the spongy, this is all trabecula. Compact is this, it's more solid here. When we look at the bone model, which we showed you a little bit earlier, I'll give you some time where you can actually pause and you can look at the key off to the right along with these landmarks. So if you want to pause it and study this, you certainly can, okay? But it's all the same landmarks that we covered in the bone histology slide earlier. Let's see, the characteristics of connective tissue. Most connective tissue is highly vascularized, with the exception of cartilage and ligaments, right? They're avascular, they're poor blood supply. Muscles, lots of blood supply because they need lots of oxygen for the mitochondria to produce energy. But cartilage and ligaments, when they get damaged, it takes a longer time for those to heal, much longer time because poor blood supply. Poor way of bringing nutrients to them if they're trying to heal themselves. And in terms of bringing wastes away, you need good blood supply to do that. So muscles heal quicker. If you strain a muscle strains can heal, but when you sprain a ligament, they take a much longer time to heal themselves. Muscle, 3 types of muscle: skeletal, cardiac, and smooth. Skeletal is voluntary. It's the only one that's voluntary. But skeletal and cardiac are both striated. Cardiac and smooth are involuntary. You can't consciously control them. Cardiac is heart. Smooth muscle is around blood vessels, the intestines, the uterus, most organs. Again, they're involuntary. When you look at skeletal muscle here, look at all the striations. That's because of the actin and myosin filaments and the light passing through. When you look at cardiac, again, striated, but the difference is highly branched. This looks like the branches of a tree with the intercalated discs. Remember, when 2 cells come together, it forms that vertical, that cross-section line called an intercalated disc. These are all the striations. Intercalated discs are only seen at cardiac muscle. And last, we have smooth muscle, non-striated. We're going to see smooth muscle on the inner part of an artery, right? Not the innermost, but actually the middle part. So when you look at an artery, you have the tunica interna, then you have a middle layer, which is the muscular layer, which makes it contract, constrict, or dilate. And then the outer layer to that is the adventitia. So smooth muscle is found around blood vessels like arteries, and they make it vasoconstrict or vasodilate. Then last but not least, we have a neuron, neural tissue. And a neuron is the only type of cell of the neural system that's electrically excitable. And it's got 3 primary moving parts. It's got dendrites, a cell body, and an axon. So here are the dendrites. It comes from the Greek word dendra, which means tree. The fingers would be my dendrites. The palm of my hand would be the cell body or the soma. My forearm would be the axon. So we have dendrites, the fingers, the branches. They feel and sense. They bring sensory information into the cell body, to the nucleus and the cell body. The cell body processes it. And then the motor information moves out through the axon. And it moves all the way to the end of the axon where we have the axon terminal. Notice the axon is covered by these myelin sheath. Myelin sheath is fat, it's lipid, it's cholesterol, it's an insulator. All the myelin sheath is if you look at this cord, we don't see the wires because it's protected by this insulation. The insulation makes the electrical conduction move so much faster to get to the end point. That's what the myelin sheath does. Wherever things are myelinated, action potentials or nerve impulses move super quick there. And then it gets to the end at the axon terminal where we have these end bulbs. And these end bulbs are stored, what's stored there are neurotransmitters. So the neurotransmitter that's there can open up through exocytosis, and maybe it's an excitatory neurotransmitter. Maybe it's going to stimulate something to happen. Or maybe it's an inhibitory neurotransmitter. Maybe it's going to stop something from happening or slow it down. So the neurotransmitters are the chemical messengers that are going to be the on switch or the off switch for whatever the axon terminal is butting up against. Okay? Okay, so now what we're going to do is we're going to close this, and we're going to look at some of these slides, these histology slides, okay? Let's bring this, let's see if I can open this. Whoop. Yikes. I got something blocking it. Maybe that'll do it. Oh no. Ah, okay, give me a second. I got a few things in the way here, and I'm trying to move them out of the way. There we go. Okay. Let's try this now. Okay, so let's look at some of the tissues here and take a look. So here is a lumen of a vein. So when you look at the inside of a vein, right here, these are one cell layer thick. These are red blood cells, but right here, this is a simple squamous cell. What lines blood vessels are always simple squamous, simple one cell layer thick for diffusion, because the red blood cells have to release oxygen. They have to release the gas that has to pass through there. So this is, if we point right here, what type of tissue is this? It's simple squamous epithelium. If we look here, if I say what type of epithelium is this? Well, this is flat, irregular cells, simple squamous cell, simple squamous epithelium, right? It's flat, irregular. If we go here and I find the lumen, you always got to find the lumen, right? Here there's no lumen, but here there is a lumen here and there's a lumen here. If we look at this lumen, we see flat cells here. So these flat cells at that apical surface is simple squamous epithelium. Now we look here. Here's the lumen once again. This is a kidney tubule. Here's a lumen, here's a lumen, here's a lumen. When you look at the lumen and you look at these cells, these aren't flat anymore. Well, they're not column, these are cube. So they're simple cuboidal cells. Here, we look at the lumen from a different cross section. These again, this is the proximal convoluted tubule. It's one cell layer thick, they're cube, simple cuboidal. We look here, here's the lumen. Again, they're not flat, these are cubes, simple cuboidal epithelium. Now we look here, this is different. Here's the apical surface. It looks like there are little hairs up here, right? These are microvilli. These are columns, but they're only one cell layer thick, so it's simple columnar epithelium. Simple columnar. These are called goblet cells. You know what goblet cells do? They secrete mucus to the microvilli to allow nutrients to stick to that mucus substance, to pull things in, or to attract debris. Here you can see exocytosis in action. Here's the goblet cell. You can see it secreting it. Isn't that wild? But these are, again, simple columnar epithelium. They're column-shaped. These are microvilli. These are column-shaped again, simple columnar epithelium. So the way I want you to study these at first is look at the name and then visualize it. Look at the name, then find it. Then after a while, look at it and see if you can figure it out yourself, okay? Pseudostratified columnar epithelium of the trachea. Well, these are column-shaped for sure. There's lots of goblet cells here. And you can see that these are some cilia or microvilli. These are cilia, but the nuclei are scattered at many, many layers. So pseudostratified columnar epithelium. A close-up look again. Pseudostratified columnar, it's actually pseudostratified ciliated columnar epithelium. These are column, these are goblet cells, these are the cilia. Again, it's of the trachea. So pseudostratified ciliated columnar epithelium. Again, pseudostratified ciliated. Okay, now we look at this one. Let me go back. Look at the top layer. Here's the lumen. Let's look at the top. Many, many layers. What are we looking at here? This is the esophagus. So food goes in the mouth, goes down the esophagus. Let's say you swallow a sharp cracker with a corner, you could scratch the epithelium. The epithelium, like the skin, needs to have many cell layers thick. So you got many cell layers of squamous. So once it's many, it's not simple, stratified. So you got stratified squamous epithelium of the esophagus. And again, it's at 40x, not 10x, not 40x. Could be 100x, but this one is 40. Here is the vaginal area. Okay, here's the lumen. Look at the apical surface. Stratified squamous, yep, they're flat, irregular cells. Not all of these, not down to the bottom, but at the apical surface for sure, flat, irregular cells. Vaginal area, yep, there could be abrasions that are going on there. There's acids there, pH issues, so you need multiple layers to protect the vaginal wall. Here's 80x, right? So like, think of it. You went from 40x, now we're zooming in on the microscope to an 80x magnification. And now you can really see the squamous cells. They're stratified squamous epithelium. Pretty cool, right? Thick skin, 40x, stratified squamous again. Just many, many layers of that stratum corneum. The stratum corneum, again, is stratified squamous epithelium. Again, here's the skin. Stratified squamous epithelium, all of this. This is probably getting ready to slough off over here. Esophagus, stratified squamous epithelium. Many, many layers. Again, look at the name and see if you can find it when you're first doing it. Stratified squamous epithelium. Again, all of this here, all of this stratified squamous, keratinized skin. All right, now we're looking at a sweat gland. Here is the lumen. Well, it's not one cell layer. We got many layers going around here. So we know it's stratified. They're cube-shaped, stratified cuboidal epithelium. Now this one's a little tricky, right? We look here, well, this one looks cube, this looks cube, this one looks columnar, this one looks a little flat, right? Different shapes. Well, it's the urinary bladder. We know the urinary bladder can expand and contract depending on if it's filled or emptied. So this is one of the few places we see transitional because they can transition these cells. Transitional epithelium. Again, urinary bladder. The cells, some are cubed, some are flat, some are a little bit wider, but this is going to be, again, transitional epithelium. This too, urinary bladder at 160x, transitional. Looks like cubed, some look a little bit longer. It looks like it has an apex and a base to it. Once you start to see different shapes, you got to go transitional. Now, this is a great picture right here. This is a muscular artery, right? So muscular artery, if this is an artery, the innermost lumen right here, this is simple squamous. It's got to be one cell layer thick because you got blood, red blood cells. They're going to have to bring oxygen to the deeper tissues. But this is smooth muscle. This is smooth muscle, not cardiac muscle, not skeletal muscle, smooth muscle. So once we see that it's an artery, what type of tissue is this? Well, it's connective tissue. Can you be more specific? It is smooth muscle. If we point here, what type of tissue is this? This is epithelium. And it is simple squamous epithelium. So in this, you have simple squamous epithelium here, but in this area, we have smooth muscle, which is connective tissue. So we're looking at epithelium and connective tissue here. Keep that in mind. This spider-like meshwork is areolar connective tissue. There's really no way of trying to help you figure it out, but they all look like this. You see some collagen fibers. It looks like a trabecula mesh-like. This is the level that kind of separates the skin from the deeper muscle layer. Again, areolar, areolar, areolar. I'm not going to ask you reticular. Wouldn't worry about that. Don't worry about reticular. Adipose, you got to know for sure. Adipose looks like simple squamous, doesn't it? Except simple squamous cells have the nuclei in the center. This one doesn't. So the nuclei are more to the periphery. They're more at the ends, if you can see them, but not in the center. Fat cells can increase in number up until the time of puberty, which is why body composition is really important to maintain up until the time of puberty. If you eat lots of junk food and eat lots of refined carbohydrates and lots of bread and lots of pasta and lots of all this sugary stuff and cakes, and you don't exercise, those fat cells that you have can increase in number, and plus they hypertrophy and they get fatter, they get larger, right? When you expend energy, then you got to burn those calories. You need fuel for that. The sugar starts to get out of those fat cells and they shrink. So that's what they say, when you're burning fat, you're actually burning the sugar and getting rid of the toxins that are stored in there. Fat cells store lots of toxins. Fat cells have really important function, which makes us look at obesity and morbid obesity in a different light. Not just that a lot of people may not be eating properly and eating the right foods, but fat cells are a storage facility for toxins. Think about it. You don't really see a morbidly obese spider, a fat ant. You don't see those, right? But think about this. You have a garden, you spray pesticides, and you have that insect on that fruit or vegetable that's now poisoned from the fungicide or herbicide or pesticide or Roundup, whatever that is. It consumes it, it gets right to its nervous system, and it paralyzes it, kills it right away. You can eat the same pesticide foods sprayed with all that junk that's so carcinogenic, and it is killing us, just killing us slowly. It's not fast killer. Why isn't it killing us fast? We have fat cells. So those toxins can go in a fat cell, and over time, they can rupture. The toxins go to the liver, and it tries to detoxify it for us. Then it sends it to the kidneys, where it gets rid of them. It makes those fat-soluble toxins water-soluble, so we can pee them out. Okay? So adipocytes or fat cells, make sure you know that identification. That is a adipocyte or fat. Okay, don't worry about dense regular. The only thing you can see about dense regular, right, it kind of looks like tendons or smooth muscle, right? Only because, like, if you look at tendons, all these collagen fibers, they're all in alignment. They're all tightly bound together, parallel in the same direction. All the collagen fibers are interwoven parallel, very tightly. But when you look at dense irregular, you can see the fibers are in all different directions, not just in one direction. So the skin, right? You don't have to massage the skin up and down. You can manipulate it in all these different directions, and it doesn't tear apart because of the dense irregular fibers. All right, when you see these raindrops, this is hyaline cartilage. Okay, these are like, can't really explain other than they look like little icicles or raindrops. That's hyaline cartilage. That's found between all of your diarthrotic joints. That's an important one to know. Again, that's hyaline cartilage, all those little raindrops with those little nuclei. I'm not going to ask you elastic cartilage. Not going to ask you fibrocartilage, not on the tissues. But look, what is this? Looks like the osteon. That's compact bone, right? Look at all those concentric lamellae. All of these are osteons, right? So this is compact bone at 40x. In the center is the Haversian canal or the central canal. This is, again, compact bone. These are the lamellae. These are the centric canals. This is a lacunae. Each of these black sections is lacunae, which houses an osteocyte, which at one point was an osteoblast that produced matrix, got hardened, and got caught in itself. Again, a close-up. This is at 80x, compact bone. Again, an osteon. There's the the central canal or the Haversian canal. This is the lacunae that houses the osteocyte. And then these are the canaliculi, those finger-like jellyfish extensions coming off of it. These are red blood cells. Now we're looking at liquid connective tissue. This is an RBC. In between is the plasma. And then this is a white blood cell. 1, 2, 3. 3 heads to the nuclei. It's granulated, that's the neutrophil. When neutrophil counts are high, you're thinking bacterial infection. Okay, so that brings that to a close. Let me stop the share here. Right, so what we covered there was 4 different types of connective tissue. We did epithelium, connective, neuron, and muscle. Muscle was smooth, cardiac, and skeleton. The neuron had the dendrites, axon, and cell body. And then we did the epithelium that had simple or stratified, cuboidal, columnar, or squamous. And then it was either simple squamous, stratified squamous, simple columnar, stratified columnar, simple cuboidal, stratified cuboidal. Then you just had to identify them. Then there was the pseudo-stratified ciliated columnar epithelium. Pseudo-stratified is false, and then transitional. Then connective tissue had all the funky stuff. All right, so make sure you review those. All right, guys, doing good so far.